Hi students, this is Professor Fernetovich, and this is a presentation for the Cannabis class at Holyoke Community College. Welcome back. Today we're going to talk about cannabis and genetics. This is in chapter 16 of the Cannabis Grow Bible, and there's a lot of information in that chapter. It's an excellent chapter. You can skip the part about the Hardy-Weinberg equilibrium for now. But the background on genes and Mendelian traits and Mendelian inheritance patterns is what's important for us. I think it's the most relatable for a beginner, and it's where we want to start our exploration. So we are going to be looking at basic Mendelian inheritance patterns, Punnett squares. You might have done this in general biology, and we'll look at some examples in different types of plants and animals as we go. I like to use some human examples because they are relatable because they apply to us, but we'll also look at some plant examples and of course some cannabis examples. So first off, where do the genetics come from? You might hear about genetics in relation to cannabis. There's a lot of breeding out there to get the best strains of cannabis and get those desirable traits. Well, the genes are found in the cells of the plant, and the reproductive cells are in the flowers. So these are the cells that are responsible for reproduction. This is a female flower shown here. This is a nice big cola with sugar leaves and fan leaves coming off of it. And over here is what the male flower looks like. The female flowers contain the female reproducing cells, and the male flowers contain the male reproducing cells, and male and female reproducing cells are both called gametes in general. So in here we've got the female gametes, over here the male gametes. Now, if you are not interested in genetics or breeding your cannabis plants, you will probably not see the male flowers. It is the female flowers that contain the cannabinoids that we're interested in for medicine and for adult use. And the male flowers contain pollen and not much cannabinoids. So the whole male plant will be a lot less potent than a female plant. And likewise, if you have male plants in your grow, you should get rid of them as soon as you notice them, because if they fertilize the female plants, then the female plants will start to produce seed, which makes a not desirable product. Once it has seed, our cannabis is no longer called sensimelia, if that's what you're looking for. And it also decreases the cannabinoid profile. So you'll have less THC and CBD and all that good stuff if you have pollination across the two. So the only time you would want male flowers is when you are doing your breeding. And you want the pollen because that contains the male gametes. The female gamete is called an ovum. It's like an egg, and sometimes you'll hear ovum, egg used interchangeably. It's really not that important of a distinction for us. And the male gamete is the sperm, same name and purpose as the human sperm. So females have the ovum, which can be fertilized by the sperm in plants, just like in animals. So both the sperm and the ovum have half the genes of the parent plant. Sperm has half the genes, the ovum has half the genes, and they have gotten that way through a process called meiosis, which we cover in general bio. It's a way of splitting up the genes so that the gametes end up being haploid. That's what it's called, to have half the genes. So when the genes combine, when the gametes combine, that is, when the sperm fertilizes the ovum, we restore the diploid state, and we go from 2 haploids to a diploid. So for humans, a human cell, like in our body, has 46 chromosomes. A sperm cell has 23, and an egg cell has 23, and when they combine, boom, you get 46 again. So the 23 and 23 would be diploid for us, and the 46 is 23 and 23 are haploid for us, and when you combine them, the 46 is diploid for us. And so the same thing happens with our cannabis plants. You have haploid parent cells from the gametes combining for a diploid individual, and that will be in the seed. This is just to highlight that haploid-diploid life cycle in humans. We could imagine cannabis plants here too, but we have female, male. Through meiosis in the sexual organs, we have sperm cells, which are haploid, and egg cells, which are haploid. They come together in fertilization, and we end up with a diploid zygote. And that will grow, reproduce those cells, double, and we end up with multicellular diploid adults. We go from 1 cell to 2 to 4 to 6, increasing exponentially until we end up with all the cells that we have. And we've got trillions of cells. We all start from 1. Every sexually reproducing organism does. So that's pretty neat. So in a plant, we would have a female plant and a male plant. We have those 2 different sexes of plants. They're dioecious. And the female plant has female flowers where the female gametes are produced, and the male plant has male flowers where the sperm cells are produced, and that's in the pollen. They come together, making a seed, and that seed will grow through mitosis, and you'll end up with the adult plants again. So let's talk about how we breed these things and how we could predict what we're going to get when we do our breeding experiments. Because you might have 2 parent plants that have great traits, and you want to know how those traits are going to be carried off through the offspring. And so that's what we're exploring here. How can we breed, and what will we get out of these plants? So crossbreeding experiments often use true breeding plants. These are called homozygous. And that means that they have been observed through many generations, and we know what to expect when they are cloned and when they reproduce. So they have certain genes, we call them homozygous. And when we choose varieties that differ in just one trait, we are doing a monohybrid cross. Mono means one, hybrid crossing. So the reciprocal crosses, Mendel performed reciprocal crosses where he was breeding pea plants. Gregor Mendel, he's talked about in the Cannabis Grow Bible a little bit. He was our founding father of genetics, and he took different pea plants, looked at true breeding plants that were tall. They always produced tall offspring. True breeding plants that always produced short offspring. He bred them together. Well, he had parent generations that are P. He called them P for parents. The first filial generation, that's the offspring from the parent generation, that's abbreviated F1. And then the second filial generation is called F2, the F2 generation. By going from P to F2, doing multiple generations there, we're looking at 3 generations total, we can determine essentially if we have a parent generation that's homozygous or not. Sometimes it's a little difficult, but we can often tell from just those 3 generations what we have. So modern genetics says that each trait in a plant is controlled by 2 alleles. These are alternate forms of a gene. We represent them by letters, uppercase and lowercase, distinguishing between 2 different types of alleles. I must say that this is the broad view, and not every trait is governed this way. So we're looking at Mendelian traits here. There are some that are blending and some that have multiple alleles that are responsible for the overall expression of the genes. But this is some basics right here. So each trait in a plant is controlled by 2 alleles. So for height, you have 2 different forms of that gene. There's a tall gene and a short gene. Well, technically we would say tall allele and a short allele in that overall trait. We have flower color there's one variation and then another variation of flower color. And the way that they interact shows us its expression, how those genes are expressed and what we see. So the alleles are dominant or recessive. We represent dominant alleles with a capital letter and recessive alleles with a lowercase letter. And the dominant allele is the allele that it's not most common in a population, but it will mask the expression of the recessive allele when it's in combination with it. So if you have one dominant and one recessive allele in an individual, one of each, the dominant allele is going to be expressed. Okay, the recessive allele gets hidden. It's still there, but you just can't see it. So we call that homozygous when you have identical alleles and heterozygous when you have two different alleles. So having a dominant and recessive allele is heterozygous. We can kind of see this showing up in humans. So for example, brown eyes are more dominant than blue eyes. And it's not as straightforward as it sounds, but I'm going to generalize here. Somebody who has brown eyes can have two alleles for brown eyes, or they can have an allele for brown eyes and an allele for blue eyes, but they show up as brown eyes. Does that make sense? My husband's that way. He has a dad who has brown eyes and a mom who has blue eyes, and he got a gene from his dad for brown, gene from his mom for blue, but because brown dominates over blue, he shows brown eyes. It's pretty cool. I have blue eyes, and that means that I must have no brown-eyed alleles. So how does this all show up? How are we going to put this together and put it down on paper? Well, we use this thing called a Punnett square. And you might think that these are just for general bio, but I've seen people use Punnett squares for crossing, cannabis, and even rabbit breeders using Punnett squares, drawing out how the genes are going to be passed down from parent to offspring to make predictions about their next generation. So this is a useful skill to have, knowing how to make and use a Punnett square. It's a table that lists all the possible genotypes resulting from a cross. And so genotypes are those letters, the alleles, represented by a capital and lowercase letter. How we make a Punnett square is we line all the possible sperm genotypes on one side of the square, and all the possible egg genotypes are lined up on the other side. And we do these at right angles. I'll show you an example in a second. And then every possible zygote genotype is placed within the squares. The zygote is the offspring. So here is a simple example showing Mendel's monohybrid crosses. I want you to take a look at this, and in my next video, I'm going to go through this and explain all the steps here. So take a few minutes, look at this, and see what you notice.